here we go. Central dogma of molecular biology. Yeah. And again, this is Francis Crick's term. 56. Yada yada yada. And here it is: DNA, RNA, protein, trait. So we talked about RNA. You may have noticed in here there's no letter T's, yeah? There's only the letter U, and that's for RNA, and we'll talk about that very shortly, yeah? Okay, so DNA to RNA to protein. The process of taking DNA information and converting it into RNA information, we call that transcription, right? RNA information being used to make proteins, that's translation. And we'll talk about each of those things individually. Right? So here it is. The traits that we have, right? The fact that I have this particular hair color is because I have a particular piece of DNA that encodes this color proteins that my cells in my scalp read, make into messenger RNA, and then that messenger RNA is turned into a protein. Does that make sense? Do you guys remember where proteins are made in cells? On ribosomes. Good, yeah. So ribosomes are where proteins get made. The DNA in my cells is in the nucleus. The messenger RNA takes the information from the nucleus out to the ribosome to be made into a protein. Yeah? Does that make sense? And again, if I have a piece of DNA for this color protein, I end up with this color hair. In other words, my DNA determines the proteins I make, which determines the traits that I have. Yeah? And that's true for me. It's true for all of us. It's true for every microbe. Yeah? Guys good? Questions? Okay. So here is the basic process. Yeah? DNA converted into messenger RNA, and then the messenger RNA goes to a ribosome, and the ribosome reads a ribbon of RNA to make, it says here, polypeptide, yeah? Do you guys understand the term polypeptide? Effectively, this means protein. It's a technical, it's a bit of legalese, right? Because sometimes a protein has quaternary structure, and it's made out of many strands, yeah? Each of these little purple balls is what? An amino acid. And you have a string of amino acids. You should have a protein unless you have multiple strings, and that's quaternary structure. You guys remember that? So that's why it says polypeptide. If you just wrote protein in here, that might be helpful, yeah? Is that clear? Does that make sense? Effectively, it's protein, or it's a piece of a protein. All right. And is this one of my cells or your cells? Right? The DNA in my cell should be where? In a nucleus, right? This doesn't show a nucleus. This is a prokaryotic cell, which doesn't have a nucleus. But the nuts and bolts at their barest level, at their lowest level, this is pretty much the way it happens, yeah? We'll talk about the differences between prokaryotes and eukaryotes eventually, but the basics are we take a piece of DNA, we make a messenger RNA out of it, a single strand out of it, and then that single strand is read by a ribosome to be made into a protein, right? If you understand this diagram, you understand something big and important, yeah? Okay. Questions? Okay, moving on. So, the universality of the DNA code makes this possible, and this is freaky. Yeah? This is something I usually dwell on fairly extensively just because it should freak you out. Have I shown you a picture like this before? I might have shown a couple pictures that are like it, but I think I usually don't show this particular. I'm going to turn out the lights. So, check it out. What is that? It's creepy, yeah? How many people are creeped out by this? How many people are fascinated by this, right? Right? So, whether you're creeped out or not, this is amazing, yeah? Right? This is mind-blowing, and this is technology, right? This is biotechnology. This is what's possible nowadays, now that we as humans, are now masters of DNA, yeah? And we've certainly not mastered the whole thing, but we are now to the point where we can do this, yeah? This is not a retouched photograph, yeah? This is not a photograph under particular trick lighting. This is a real tobacco plant, yeah? Tobacco. The stuff cigarettes are made out of, yeah? And that tobacco plant is giving off light. It is blankety-blankin' glowing, yeah? Right? And why is it glowing? Am I stealing your thunder here? I don't know what to say, yeah? No. Bioluminescence, that's just one part. All right, don't scare me like that. That wouldn't be the first time, but yeah, so I'm stealing your thunder too, yeah? All right, so to some degree, right? This happens naturally, right? How is a firefly? You guys know what a firefly is, yeah? You don't know what a firefly is. All right, so you guys this summer need to go camping, yeah? Because a lightning bug. Do you know what a lightning bug is? What about you, Katie? Are you good now? You guys all know what one is, yeah. Okay. Have you seen one before? You've never seen one. So you need to go camping. What about you guys? You catch them and put them in a bottle, yeah? What about you, Katie? You guys should still go camping, but you don't need to go camping in order to see one because you've seen one, yeah? Is that clear? But Xiao, you're under obligation, right? You need to see one of these things in your life for real, right? And Massachusetts, they have them in the summertime. You'll see them, yeah? Or at least in western Massachusetts, and I've seen them in Connecticut, and I've seen them twice in my life, and both of the times were here on the East Coast. I've never seen them anywhere else. Okay, so how do we make this plant? Yeah, Netta. Gotta stand closer so you can talk into the microphone, please.
This plant is genetically modified, so it means so the DNA from like the
firefly, called a firefly.
Some species in the deep down the ocean, they when they live in a
That's a jellyfish. We'll show that in the next couple slides. Yeah, go on.
They get the DNA from that and they put it in their DNA, so they make it their own. So it's not something that naturally happened for this plant. So this is after modification happened.
Did you guys hear that? Very, very astute and eloquent description. Yeah. So take a piece of DNA, put it into get a firefly, right? So you're Xiao, yeah? You want to make this plant? What do? First step, go camping, yeah? Step 2, get a jar. Step 3, grab a lightning bug, yeah? Grab a firefly, right? Step 4, take them home and squish them up, yeah? Step 5, add some chemicals, detergent, alcohol, etc., and purify out his DNA. Step 6, get a piece of molecular scissors and cut out the piece of DNA that encodes the gene that makes that firefly glow. Yeah? The reason the firefly glows is because the firefly has firefly DNA. Yeah? And that firefly DNA encodes proteins that can take a chemical substrate and convert it into a chemical that gives off light. Yeah? A glowing chemical. Just like those little Sialum, you break the little plastic glow stick, yeah, and then the chemicals mix together, and the chemical reaction gives off light, yeah? You have fireflies, you have an enzyme that pretty much does a pretty similar thing, yeah? Okay, so, and that enzyme is called luciferase, right? Why is that? Is it because it's evil? Right? Metal! No. It's not, the name comes from bringer of light, yeah? and it's an enzyme that brings light, yeah? And if you've got the DNA to make that enzyme, and you have the DNA for the chemical substrate, then you can make that light, yeah? And if you are a tobacco plant, do you normally, as Neda said, do you normally have that piece of DNA? No. Some hotshot scientist purified that piece of DNA out of a firefly and then stuck it into your seed when you were just a little seedling growing, yeah? Right? And then your seedling cell divided into 2 cells, and then 4, and then 8. And every one of those cells had the little piece of firefly DNA. And so the little piece of firefly DNA got copied from one cell to the next, to the next, to the next. And every cell in that little guy is now got a piece of firefly DNA. Yeah? And why is it giving off light? Because fireflies and tobacco plants both use DNA, and they both read the DNA in the same way, right? If the firefly has AAG, right? Where's A? There's A, yeah? Where the heck, this thing never... AAG. If the firefly has AGG, it makes a lysine. And if you give that piece of DNA to the tobacco plant, and the tobacco plant sees AAG, it makes lysine too, right? Because, again, they both read the DNA in the exact same way. They both obey this codon chart. This codon chart is true for both the firefly and the tobacco plant, yeah? Right? Which is remarkable, and again, it allows this remarkable and crazy technology. This mind-blowing technology, yeah? Amazing, yeah? Could you smoke this? This is a tobacco plant, yeah? Could you dry out the leaves from this guy and shred them and put them in a cigarette and smoke them? Yeah. Would it taste the same? I don't know. It might. It might taste a little weird because there's fluorescent chemicals in there. Maybe those chemicals have a taste. Could you still get cancer? Yes, you still could get cancer smoking tobacco, whether it has firefly genes in it or not, is harmful to your health, yeah? Does that make sense? Is this remarkable? Why do they do this? Right? Is the whole purpose of this technology just to freak out community college microbiology students? Right? So the answer is certainly no. Yeah? The reason they do that is partly, right, as far as why this tobacco plant was made, right? Well, conceivably, you could use these guys to light up your walkway up to your house, right? Instead of those little solar things that you stick into the, right, and somebody always steals them because they're not nailed down. You guys know what I'm talking about? Yeah. Instead of those, you could plant these guys, yeah? And you could find your way to your front door using glowing tobacco plants, yeah? But maybe that doesn't sound very practical, yeah? That's probably not necessarily enough to justify this as more than just an entertaining parlor trick, yeah? If you lit your walkway with glowing tobacco plants, that would be great as people came to your house for a party, right? But as far as making the world a better place, is that really happening? Right? So what about this? Here's another gene, GFP, right? What's the code for GFP? Well, there's the protein, yeah? This protein is a light-trapping protein, yeah? And I'm sure both of you guys will be talking about it in your presentations. I hope not to steal too much of your thunder right now, but I want to talk about it briefly, right? So there's the sequence. There's A, G, there's every letter. If you give this piece of DNA that has an A, T, G, G, C, T, T, C, C, yada, 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 right? You get that from this jellyfish, and you give it to this worm. What does the worm do? It becomes green and fluorescent like the jellyfish. The jellyfish Achoria victoria, which which you can find floating in the water off of the coast of Vancouver. Yeah, this is found in the Pacific Northwest. Victoria is a town near Vancouver, actually. In any case, this guy, squish him up, get his DNA, put it in this worm, and what do you see? You see this green light and this is not giving off light. This guy is sort of standing under a fluorescent light. It's a black light. Do you guys know what a black light is? Right? They have them at the disco. Yeah. Right? And it makes your white T-shirt look purple. You guys know what I'm talking about?
Yeah.
And then you have the fluorescent paints that look normal, but then they glow crazy luminescent, right? Because those are not giving off light. They are they're absorbing light and releasing it. Yeah, does that make sense? So this guy what part of him is absorbing light and releasing it? What's this green part here? This is a worm called C. elegans. This C. elegans worm is a very important model organism, right? Biologists, sometimes they do work on mice, yeah? Is that true? That's his brain. That is correct. So this glowing part here is a worm's brain, yeah? What about these other lines going off here and along there and so on? You see those? What are those? Those are the rest of his nervous system. Does it matter if we can light up the brain of a worm? We can learn about the architecture of their brain, yeah? What else? Well, we could conceivably, what if we had a headache medicine, yeah? Is that medicine better than aspirin? Is it safe to take, yeah? Well, we could take that headache medicine and sprinkle it on these guys as they were growing up, and if we saw that their brains were all dim, whereas ones that didn't grow up around the headache medicine were brighter, what would you think? Maybe we wouldn't want to take that medicine. Does that make sense? Yeah. Right? So again, that's pretty important, right? Do you know how big these worms are? They're about 1 millimeter long. They're really tiny, right? So for every mouse that we grow in a scientific laboratory and do experiments in a scientific laboratory, we could do experiments on thousands of these worms, yeah? And maybe we do fewer experiments on mice. Who do you care about more, mice or worms? Mice, yeah? They're more adorable, they're fuzzier, right? What do you think? So, to some degree, I'm jabbing you guys, right? Does that make them more virtuous? Right? Certainly, I feel better doing horrible things to bacteria in this class than I would feel if I were doing horrible things to mice, yeah? Does that mean that mice research is something we should just forget about? Certainly not, right? We get real knowledge out of doing horrible things to mice, yeah? But it would be nicer if we could do horrible things to worms instead, don't you think? I would feel better if we could do horrible things to bacteria instead of worms. And I would feel better if we could do horrible things to worms instead of mice. Does that make sense? Right? Okay. So, and yet, here we go. Yeah? There's mice. Glowing green. What do you think? So again, he's got the same piece of DNA. We can take it from the jellyfish and give it to a worm, a bacterium, or a mouse, and it'll glow, right? Which mice have been genetically modified? Looks like mouse 1 and mouse 3, right? They've got glowing green tails, glowing ears, glowing noses, glowing eyes. This guy looks pretty normal in between them, yeah? And so, again, if these two mice go to the little mouse disco, what's going to happen? Well, all the lady mice are going to be like, whoa, now that guy's glowing. Maybe. Speculation, yeah? But again, can we see the architecture of a worm's brain? Yeah? Is that worth knowing? It is definitely worth knowing. This technology got a Nobel Prize pretty recently, yeah? And it didn't get a Nobel Prize for its power to entertain community college students, right? It got its Nobel Prize for its power to do real things, yeah? Here is change GFP's DNA. You can change the color, right? You change the DNA for GFP, right? And then suddenly you're no longer making GFP. You're making YFP, or you're making BFP, blue fluorescent protein, or CFP, cyan, that's like a turquoise color, yeah? Does that make sense? Look around in nature for a few other proteins, and then you can get all these other colors too, right? Finagle those proteins a little bit, and you can change those colors, right? This one is derived, all of these ones are derived from GFP. All of these are derived from another fluorescent protein, yeah? And again, take all these pieces of DNA and put them into this fish, and what do we see? We see every neuron in this fish, in the fish's brain, in the fish's entire nervous system. This is called the brainbow, right? Appropriate, don't you think? Is this important? Are there some similarities between the brain of a fish and the brain of a person? Sure, some at the basic level, yeah. And we can learn in detail about the fine points of the architecture of a fish's brain by lighting up every one of its neurons, yeah? And if we knew how the brain of a fish was built, we would know how the brain of a person was built, yeah? And again, right, maybe we could find interneural connection-based diseases, maybe schizophrenia, maybe another disease, right? Wouldn't it be nice if we could make some progress against diseases like that? And certainly, if we knew about the structure of the brain, we would be better able to make that progress, yeah? Don't you think? Again, this is not just, it's awesome, right? It's fascinating, but it's not just a parlor trick, right? There are real, critical, and very powerful reasons for this technology. Yeah? Okay. But it is, at some level, there is no denying that it's just crazy cool, isn't it? This is like, whoa. Does that mean it's morally okay? What do you guys think? Raise your hand if this is okay. Raise your hand, this is not okay. Raise your hand, how the heck should I know? Right? So again, hopefully you won't get an answer from me, right? Every person gets the right to make moral judgments about the world that they live in, yeah? But again, this is technology, and whether this technology is okay or not is separate from whether it's cool or not, yeah? I have my own opinions about whether it's okay. I think whether it's cool or not is not debatable. This is cool, even if it's not okay. Don't you understand the difference there? Right? It's fascinating. I personally have my own views about jet planes and stealth bombers and so on, but that doesn't mean that I don't think that they're cool, right? They're fast sports cars, right? Those are cool, yeah? I don't know if I'm making sense here. Okay. And again, whether they're okay or not is a completely different question, yeah? And one other thing to point out is that it's a different question when you're talking about finagling a worm versus a fish, yeah? And it's a different question whether you're finagling a fish versus a mouse, yeah? And it's a different question whether you're finagling a mouse versus a plant versus a person. Do we have the technology to do this to people? What do you think? Yeah, so a human is not much more molecularly and cellularly and genetically complex than a person, yeah? We're smarter, right? We're bigger, but we're not necessarily more complex on the cellular level, yeah? Can we do this to people? Do we do this to people? We definitely do not. And we've passed laws that say that finagling human DNA is illegal, yeah? So And yet, sorry.
Okay, can we talk about it later because it's our presentation?
Oh yeah, I'm stealing your thunder. Okay, all right. Okay. All right, so we'll continue this later. Yeah.